this will be the first portion of glycolysis. We'll just start at the beginning. We'll see how far we can get at the end of this video, and then that'll determine where we are and what will be on the next video. But the next set of videos will detail glycolysis. And so glycolysis is all about this molecule. And this is glucose. And so glycolysis is all about glucose and the breaking down of glucose and getting energy from that breakdown. And so one of the things that we're going to look at is obviously how this glucose is broken down, the different steps that it takes, the different enzymes, how those enzymes are regulated. They will all be very important. The first step, so glucose gets imported by a GLUT, the glucose transporter, into the cell. Once it's in the cell, there are two enzymes, depending on what cell it goes into. Hexokinase are in muscles, or glucokinase, which is in the liver. And what we actually see with hexokinase is that the Km of this enzyme is about 0.1 millimolar, whereas glucokinase, the Km, is about 5 millimolar. And so what this tells us is that every time in the muscle or in any other cell with hexokinase, that any time even one molecule or very few molecules of glucose get in, they're going to go through this pathway. Whereas glucokinase, you can allow the glucose to build up a little bit in your liver before the glucose will go through glycolysis. And so the first step, the glucose gets into the cell. The first step is through these kinases. Now, if we remember what a kinase does, a kinase puts on a phosphorylation group. And it does it at the 6th position. Oops. Let's go and erase that. And it's done at the 6th position. So what we make is glucose 6-phosphate. I'm going to abbreviate phosphates as that from here to the end. You should be able to draw a phosphate group because on certain quizzes or exams I might ask you to draw the phosphate out, and so this is what it would be. So here's glucose 6-phosphate. Glucose 6-phosphate is a product inhibitor for those cells that do not produce glycogen. So if a cell does not produce glycogen, then the buildup of glucose 6-phosphate will inhibit hexokinase or glucokinase or any of those glycolysis enzymes that are upstream. If a cell, like the liver or the muscle, can perform glycogen synthesis, glucose 6-phosphate then goes into glycogen synthesis. We're not going to talk about glycogen synthesis because it's a little bit different than glycogen metabolism, but just know that glucose 6-phosphate can either go into glycolysis, which is what we're going to talk about now, or it can go into glycogen synthesis. So now we have glucose 6-phosphate through an enzyme called phosphoglucose isomerase. Now in some books or in some places you read this enzyme is known as phosphoglucoisomerase. Either phosphogluco or phosphoglucose, both of them are correct. And what we do is, since it's an isomerase, it's going to change the way these things are bound. And you're going to make this molecule. So again, I'm going to abbreviate the phosphates like this. However, you should be able to draw a phosphate group on these different compounds. And so what we make is this. And what this is fructose 6-phosphate. So glucose through hexokinase or glucokinase goes to glucose 6-phosphate through phosphoglucose. So depending on the energy needs of the cells, we'll talk about how we can determine that. It'll either go into glycogen synthesis or it'll go down through glycolysis. If it goes through glycolysis, it'll go through phosphoglucose isomerase, which will turn it into fructose 6-phosphate. Now, what's interesting to note about these two different products is the delta G naught prime of the reverse reaction of going from fructose 6-phosphate to glucose 6-phosphate, because it is a reversible reaction because it's just an isomerase. The delta G naught prime is -8 kcal per mole. Whereas the forward reaction, I'll do it in black, the delta G naught prime is equal to -0.6 kcal per mole. And so just by looking at energetics, it would suggest that glucose 6-phosphate does not want to turn into fructose 6-phosphate, right? Because energetically it's best to do a -8 kcal per mole than a -0.6. So, how in the world can we drive this reaction forward, right? Because we want to go to the end of glycolysis. We don't want it to stop here and just make a bunch of glucose 6-phosphate. The way we do it is we take the fructose 6-phosphate and we remove this product. If we remove the product of this reaction, then we drive equilibrium forward. We drive the reaction forward. Which is why the next, to remove the product quickly, we have to have a very quick enzyme on the next step. And of course we do. And so what we left off, we had this. I'm trying to draw these as clear as possible. If you have any questions about any of my drawings, you can either see me or you can reference your book, because your book should have all of the same drawings. Right, so we have fructose 6-phosphate, and it goes through an enzyme called, called phosphofructo, right, because we're using a phosphorylated fructose. So phosphofructo kinase 1, also known as PFKI. And so what PFK1 does is it uses another ATP. So again, this is the second time we will have used an energy input. H+. This will be the second time we've used energy input in this cycle. And that will be very important. And so what we make is this. So whether you draw the oxygens connected to the phosphate or your shorthand is just connecting to the phosphate, you just know that there's those oxygens there. And this is fructose 1,6-bis phosphate. And so because of this, because that we want to drive equilibrium forward, fructose or phosphofructokinase 1 is the rate limiting step in glycolysis. So rate limiting step. It is the most highly regulated step. It is the most highly regulated step in glycolysis for that very reason. So what we're going to do is we're going to kind of we're just going to look now at PFK1 because it's that important. So how is PFK1 regulated? Right? Because we said that it's the most highly regulated enzyme in this pathway. And so the first way that it's regulated is through the concentration of ATP. And so high ATP, I'll do this in red. High ATP is equal to decreased activity. And so that would be a negative regulator. So high ATP, lots of ATP is a negative regulator for phosphofructokinase 1. Why? Because if you have a lot of ATP, you're in a high energy state. You don't need to do metabolism. You don't need to do glycolysis. And so if we actually look at this, where we're looking at velocity of PFK1, and we look at the concentration of fructose 6-phosphate under low ATP conditions, you see a normal Michaelis-Menten graph. However, under high ATP conditions, you see this. You see so this is high. You see an allosteric effect. So high ATP leads to a decrease in affinity of PFK1 substrate. We honestly don't know why, and it's still being searched out, but just know that an increase in ATP, high ATP, leads to a decrease in activity of PFK1. The second regulator of PFK1 is the concentration of AMP. If you have a high concentration of AMP, that means you're going to positively regulate the PFK1. You're going to increase it. AMP reverses ATP regulation. Again, we don't know how, but we just think that because of AMP, if you have high AMP, you know you're in a low energy state, you need to run metabolism. And so, why not, right, so we're using AMP. Why not use this? Right, why not use the concentration of ADP? Because if the cell, so let's say the cell is using lots of ATP. It's growing, it's getting ready to divide for a variety of different reasons. What you can have is ADP combining with ADP through an enzyme called adenylate phosphate. And what you get is ATP plus AMP. So actually, there's a lot of talk about ADP and what ADP means in the cell. And actually what we find out is that the cells don't really use ADP much as a regulator. But in reality, what they use is they use this to tell them, this ratio, to tell them what type of energy needs the cell needs. Right? So the ratio of ATP to ADP is, or ATP, excuse me, to AMP is very, very important. So the first way PFK1 is regulated is through ATP. The second way is through AMP. And I told you guys this is the most highly regulated enzyme that's out there, or at least in glycolysis. And the concentration of citrate is also a regulator. So a high level of citrate is going to equal a decrease in activity. So it's going to be a negative modulator. It's going to be a negative regulator. So what is citrate? Here's citrate. We haven't covered the citric acid cycle yet, but when we do, you will see citrate again. And so citrate is actually in, like I said, the citric acid cycle. It's a downstream product. And so if you have a lot of citrate, then that means you have a lot of metabolism already going on. If you have a lot of metabolism going on, then you're in a high energy state. If you're in a high energy state, you don't need more metabolism to occur. And so that's why an increase in citrate is actually a negative modulator of PFK1. Now, we know that citrate plus ATP, right? Because we said high concentrations of ATP is a negative modulator. They actually enhance each other. But again, we don't know why. We have no idea. Maybe it's some sort, it has to do with how the enzyme is. We just don't know. So, the other way is fructose. Fructose 2,6 bisphosphate. I know what you're thinking. Fructose 2,6 bisphosphate. What's that? That's a separate fructose molecule that is used in other forms, in other metabolic pathways, but a high level of fructose 2,6 bisphosphate is going to increase PFK1 activity. And so it's a positive modulator. And so fructose 2,6 bisphosphate is another intermediate of a variety of pathways, and you usually see this in your liver. So this usually occurs in your liver. If you have a high amount, if you have a high amount of glucose, it's turned into fructose 6, phosphate. And so fructose 6 phosphate, if there's enough of it, can activate this different pathway to form this. And so it's very similar to fructose 1,6 bisphosphate, but instead it's fructose 2,6 bisphosphate. And so what we actually see is that enough, so 1 micromolar of this compound is enough to reverse the inhibitory effect of ATP. And not only does it inhibit the effect, it increases the affinity of PFK1 for its substrate. So, why? Right? So how is this, fructose 2,6 bisphosphate made? And so what happens is in the liver. So let's take a look at the liver. In your liver, in the cell membrane, there are two different types of membrane-bound proteins. One is a receptor, the other one is a cyclase. This is a glucagon receptor. This is adenylate cyclase. Adenylate cyclase we've seen before turns ATP into cyclic AMP. And so in low blood, so you have low blood glucose. Right? So in low blood glucose, what we do is glucagon will bind onto the receptor. Right? So this is in the opposite. So we're talking about regulating PFK1, right? So under low blood glucose, we really don't want glycolysis, at least to really occur in the liver. We want glycogen metabolism too. We want to break down that glycogen, but we don't really want it to go into glycolysis. So when glucagon binds, it sends a signal over to adenylate cyclase. Adenylate cyclase then takes ATP and makes it into cyclic AMP. And as we've learned before, cyclic AMP then activates protein kinase A. So, within our liver, there's a complex of proteins. On the left in the box, we have F, B, P, ACE. Instead of 1, we're going to talk about 2. So, fructose phosphatase 2, fructose phosphatase 2. In the middle of these proteins, there's a hydroxyl R group. On the right side, instead of PFK1, you have PFK2. So under low blood glucose levels, what you do is the pKa comes in, removes that hydroxyl, puts a phosphate on that R group. When it does that, this becomes active. This becomes inactive. So if the phosphatase is active, what it's going to do is it's going to take fructose 2,6 bisphosphate, and with a little bit of water, it's going to remove a phosphate group, and you're going to get fructose. Whoops. You're going to get fructose 6 phosphate back. Right, so we're going to get rid of that signal to say, hey, we want glycolysis to occur. That's under low blood glucose levels. But let's say we're under high blood glucose levels. So under high blood glucose levels, glucagon is not there. We use insulin. We talked about insulin, adding more glutes to different things. So if we're under a high blood glucose level, or under normal conditions, we don't have that phosphorylation from pKa, because there's no signal to activate it. If there's no signal from pKa, then the fructose bisphosphatase, is inactive, whereas PFK2 is active. And if PFK2 is active, it's going to turn fructose 6 phosphate into fructose 2,6 bisphosphate. It's going to use ATP to do that. And so fructose 2,6 bisphosphate will then go on to do what it does, activating PFK1. Now, we have to talk about something else before we can even continue on to glycolysis. So this is all the first 3 steps in glycolysis. This is how regulated this is. This is why this is very, very important. And so what we're going to do is we're going to talk about what's known as the Cori cycle. Because fructose bisphosphatase 2 plays a very important role in what's known as the Cori cycle. And so in our muscle cell, so we're going to look at 2 cells. On the left, we're going to have a muscle cell. On the right, we're going to have a liver cell. So in the muscle cell, you get glucose. Glucose turns into pyruvate. through glycolysis. Pyruvate, under anaerobic conditions, so low oxygen conditions in our muscle, turns into lactic acid. This lactic acid is then dumped into our blood. So this is the blood. If we drop a bunch of lactic acid into our into the bloodstream, it's going to decrease the pH. We have to think about what happens at decreased pH with hemoglobin. That the oxygen gets dumped then into the muscles. Well, the lactic acid doesn't stay in the blood, but what it does is it moves into the liver, where through the liver it gets turned into pyruvate, which then turns in to glucose, right? And this here, gluconeogenesis. So then the glucose from the liver gets dumped into the blood and it can go into the muscle. So that's known as the Cori cycle. So let's look at it in a little bit more detail. Let's just focus in on a couple steps, especially within the liver with gluconeogenesis. So we have fructose 6-phosphate. So in glycolysis, PFKI phosphorylates it to fructose 1,6-bisphosphate, right? And then we go down to glycolysis. Let's try that again. It goes into glycolysis. Then, what fructose 1,6-bisphosphate can do in the liver is there's an enzyme, fructose bis phosphatase 1, which, again, H2O, can remove a phosphate group, allowing us to make fructose 6-phosphate again. That can then go to glucose 6-phosphate and then make glycogen or other things. And so fructose 2,6-bisphosphate is important because it activates this. Whoops, I don't know why I did that. Let's do it in green. So it activates this, and it deactivates that. So that's fructose 2,6-bisphosphate. Activates PFKI like we saw before, but it actually deactivates FBPase1. We talked about it in class with bumblebees. Their thoracic temperature is 30 degrees Celsius at all times, no matter if it's 10 degrees Celsius outside. And so what bumblebees can do is they can inhibit this regulatory control, causing this cycle to constantly spin around, right? It's known as a futile cycle. There is no end. It just constantly spins. And as it constantly spins, it's just generating heat to keep that bee's thoracic temperature at around 30 degrees Celsius. And so we're going to go ahead and end here. So we've talked about the first 3 steps of glycolysis. We've talked about the regulation of phosphofructokinase 1, the full regulation of it. We talked about the Cori cycle. You're going to have to know each one of these. And so when we pick it up in the next video, we'll look at what happens to fructose 1,6-bisphosphate as it continues down the path of glycolysis.